Hello, everybody. This is Dr. Dan Guerra from Guerra Med Studios in the Pacific Northwest. Last time we talked about how fatty acid metabolism, particularly de novo fatty acid synthesis and the desaturation of those fatty acids, were central in metabolism and also in signaling and in cell differentiation, and even I hinted at cell fate. I said that I wanted to lay that as a foundational groundwork so that we could start using that knowledge to probe into the literature and examine how fatty acids, in particular, affect human health, and how there's a great deal of literature out there that needs to be carefully sifted through so that we don't make mistakes or errors and a misunderstanding about how fatty acids in particular either play a positive role or a detrimental role, or indeed are actually involved in the total regulation of human health throughout life, which is my basic thesis. So besides the fact that I'm biased because I'm a lipid biochemist, I'm hoping to be able to demonstrate that to you in these few lectures that we're doing here now as we're closing in on Christmas 2017. So today is actually the 7th of December, 2017. I'm going to talk to you about one important paper that came out, and then a couple of correlative papers that will help us look at whether or not the main one we're examining is indeed paradigmatic for what the literature is looking at. And I try to generate some dialectic so that we can, at the end, superimpose the current literature with what has already been published and what may be examined in the future in terms of generating hypothetical deductions and getting at new research. So let's get started after that rather windy introduction. Okay, so that's me, Dr. Dan Guerra, and that's my personal email address, which you can contact when you want to discuss any of these lectures that you see or anything about Guerra Med. That's also my Facebook page down there. I haven't posted that yet on my previous presentations, but I thought that might not be a bad idea. In the middle here is actually our website for the company and also our official email address for asking us for consultation. So again, today I'm going to talk about acyl lipid metabolism and cell fate, and the subtitle is Lipids in Human Health, and this is going to be basically lecture video 2 on this really important arc of literature. So let's get started. Okay, so I want to first of all lay some basic groundwork about what we're talking about and remind everyone that we have discussed various aspects of cell fate in previous video lectures that I presented. So I encourage you to go back to those. And if you can't find them, please go ahead and email me or just go to my Facebook and you will see where I posted them on Facebook. But if you still have a difficult time finding them, please contact me directly and I will send you the files. So first of all, let's look at this paper from Cell Death and Differentiation 2012. Here we see that during stress response, cells will interrupt the cell cycle and they will induce the all-important p53 tumor suppressor gene. Remember, that is one of those gatekeepers or cell checkpoint proteins, so that when that p53 is expressed, it alters the cell fate. That is, it alters the cell cycle. So it does it a lot of different ways. But one of the things it does, it instantiates a modification of gene expression. And some of the genes that it's altering are genes involved actually in metabolism. So one of the metabolic changes that occurs is a switch from glycolysis, which is one utilization of cellular glucose, to the oxidative pentose phosphate shunt. And when we move glucose into the OPP pathway, basically, we're going to make pentose sugars like ribose and ribulose 5-phosphate, but we're also going to make the all-important NADPH. And NADPH is actually the biological reductant in the cell. So when a cell goes into stress, it's going to, one of the things is cells that are aerobic encounter during stress is a lot of reactive oxygen. So in order to quench that reactive oxygen and get it out of the cell, NADPH is there to reduce those partially reduced forms of oxygen, which are the ROS, or reactive oxygen species. And that's exactly why the NADPH is being generated here and why glucose is being facilitated into the OPP pathway. Now, you don't just lose the glucose there, that is the carbon from the glucose, because you can shuttle back after just losing one carbon via the decarboxylase reaction at the beginning. Shuttle back most of that carbon, 5 of the 6 carbons from glucose, back into glycolysis later down the pathway. And so you don't really lose that carbon, and you can still make ATP from it. All right. So another thing that cell stress does, it leads to the potential for DNA damage. And we've talked about this again repeatedly in numerous Vera Med video lectures on YouTube here. Eventually what happens in this process is when DNA damage is detected, anabolic pathways like protein synthesis and lipogenesis, that is the de novo synthesis of lipids like fatty acids and isoprene lipids like cholesterol, that's shut down. And the cell cycle also is halted at specific checkpoints. And we've gone over that as well. That's not what we're going to do today. The failure to activate, however, the AKT-mTOR pathway is sort of the subtle backdoor, trapdoor way of regulating all this, because what happens is P53 mediates the transcriptional activation of negative regulators of the AKT-mTOR pathway, and that's the anabolic pathway. So those two of the very common negative regulators that we look at are P10 and the TSC2. I'm not going to talk about those today. I'm trying to give you a full measure of understanding of what's going on when we say that cell cycles interrupted. The reason I'm laying this as groundwork this afternoon, is because I want you to see how lipids are going to play a major role here, okay? And because it's going to impact on that metabolic pathway, and metabolic pathways are all interconnected with lipid metabolism. So what happens with this process is you initiate cell fate measures, and there are two possible things to do during DNA damage. So the senescent process, or the aging process, which prevents cells from replicating. So you shut down mitosis. That's, of course, going to be indirectly related to the cell cycle interruption. Or you can actually drive those cells directly into programmed cell death, or apoptosis. There's another pathway that can be mitigated, and that, of course, is autophagy. And autophagy can occur both in senescence and alternatively during early stages of apoptosis. Autophagy we've gone over also in the past, and I'm not going to do that today. I want you to get an idea of how cell fate is regulated by these metabolic pathways. So in a recent paper examining the molecular genetics of senescence now, and this was a paper published this year in late March, it suggested that the compound NAD, which is nicotinamide adenine dinucleotide, in the oxidized form, that's what that plus sign means, actually when you get NAD buildup, it enhances DNA repair, and it does so by preventing an inhibitory protein from binding to the erstwhile DNA repair enzyme complex. So it was also previously shown, that's one way that NAD does this, that NAD actually activates sirtuins. And sirtuins are those enzymes that are deacetylases. They remove acetate, covalently bound acetate, from things like proteins. Some of the proteins can be free enzymes in the cytosol, as we'll see later, or they can be the histones associated with chromatin. And when that occurs, when sirtuin deacetylases are turned on by NAD in the natural process of aging, that it causes senescence of the cells. Okay? So that's because condensed chromatin is a really poor substrate for gene expression. So condensed, imagine it being a collapsed structure, a physical structure gets collapsed because the acetate opens up that DNA. And when you have a collapsed structure, you don't get good gene expression, except specific genes continue to be expressed. Which ones? For example, P53, P10, proteins like that. So what can we conclude from this brief delving into senescence is that there's a limitation of gene expression, and that can trigger cellular fuel and therefore bioenergetic alterations. And that's going to lead to less cell division and more cell maintenance and repair. Now, again, remember I told you before that part of that maintenance can be autophagy. That is the digestion of macromolecules within a given cell, and then the utilization of those monomeric units, such as free amino acids, to resynthesize other proteins, so as to maintain life without necessarily being anabolic or generating any new macromolecular structures. Certainly no DNA synthesis or de novo lipid synthesis. And of course, it allows for repair during the process of maintenance. Okay. All right. So where's lipid involved in all this? Of course, lipid is a major player in bioenergetics, right? So you have two major sources of energy. You've got glucose and you've got fatty acids. You also have amino acids, such as glutamate and glutamine. Those are common ones. And those are generated, of course, by limited proteolysis of proteins in the cell. Now, they contribute variably. And the variation in their contribution to bioenergetics is due to the fact that often amino acid catabolism is linked to, is coincidental with, the elimination of ammonia via the urea cycle. So protein turnover requires that amino group be turned over, and you can do that with transamination reactions, something else I talked about previously. Or sometimes, depending on what system you're looking at, for example, liver, kidneys, that sort of system, then you actually are involved in ureagenesis, and you are removing or eliminating that ammonia. So remember that you have to pay the consequence of using amino acids if you're going to use them for bioenergetics, because you're ultimately going to have to break down. You have to oxidize that carbon, and that means you're going to have to eliminate the amino group. Because remember, amino acids have an alpha amino group. And sometimes they have more amino groups in their R group. All right. Now, glucose oxidation can occur, and we've talked about this many times, and I know those of you with any kind of cellular background, biochemical background, including you physicians, know that glucose can be oxidized anaerobically via glycolysis or completely aerobically, and that is going to require, of course, the mitochondria. And the mitochondria houses the tricarboxylic acid cycle, electrotransport chain, and oxidative phosphorylation. So obviously that's going to require molecular oxygen, because the molecular oxygen, remember, gets reduced by the very hydrogen that was originally used to reduce the NAD that was involved in the oxidation of the glucose, right, via glycolysis to pyruvate to acetyl-CoA and oxaloacetic acid, et cetera, through the TCA cycle via the dehydrogenases in that cycle. Now, fatty acid oxidation doesn't have the wherewithal to do anything anaerobically. Fatty acid oxidation, actually, that oxidation requires molecular oxygen. So if you don't have molecular oxygen, you're not going to get any fatty acid oxidation. So that ultimately and absolutely requires mitochondria, and also the peroxisome that carries out some of this. So remember, the amino acid bioenergetics, hopefully I've explained this to you already on the slide, that's going to pretty much follow glucose subdivisions in terms of how it gets metabolized. And that's because amino acid bioenergetics just simply means once you eliminate the amino group or you transfer it to another amino acid via transamination, you're basically in the TCA cycle. And so you're going to be following carbon just through that system as you would carbon coming from glucose. So now that's one thing. Lipid metabolism is really important in bioenergetics. Just again, I'm laying the foundation here of why it's so significant in cell fate. This is why you have to build up from a common foundation, right? And we have to make sure that foundation is coherent with what we understand. So if you get foundationalism, coherentism, and you go out and you find that every time you look for it, then that generates some probability that you're looking at true statements relative or vis-a-vis to metabolism and biology. So now lipids don't just end there with bioenergetics. Oh, heck no. They're also major involved in cell signaling. I mean, when I think about lipids, I think more about cell signaling than I do probably about bioenergetics. And it's just because I'm weird and I'm a lipid biochemist. But anyways, about cell signaling. Fatty acids can be oxidized to these compounds known as eicosanoids. Now, eicosa means 20. And so autocode eicosanoids are basically oxygenated fatty acids of the C20 length that act as autochord hormones. That is, they're utilized in the cell they're made in, or sometimes they can act as paracrine. That is, adjacent cells can utilize these as hormones. That is, regulators of metabolism, regulators even of gene expression. Big deal in inflammatory responses, for example. So fatty acids can be oxidized autocotinoids. And those are the cyclooxygenase, lipoxygenase, P450, monooxygenase pathways, of course, making prostaglandins, leukotrienes, and thromboxanes and prostacyclins. Okay. All right. Now, besides the fatty acids, sterile structured lipids. Remember, you got two major classes of lipids. And the linear retinoid isoprenoids, those are both of isoprenoid backbone, those can act as endocrine hormones, as we know. Okay, so we think about cortisol, we think about testosterone, we think about estrogen. Okay, those are your typical cholesterol-derived sterile endocrine hormones. But you also have the retinoid isoprenoids, such as retinoic acid, which is a really important hormone in the body. And of course, we don't synthesize that particular isoprenoid. We get that from the diet as vitamin A, from carotenoids, from plants. So fatty acids also are components. So that's another major thing about lipids involved in cell signaling. You've got all this, signaling phenomena going on with these sterile compounds, and you got the signaling going on with eicosanoids. That's not enough. That's only a small part of the picture. Included in this also are fatty acids as components of complex lipids. Which kind of complex lipids? Those containing glycerol or sphingosine. So you have glycerol phospholipids and sphingophospholipid. They mediate big time cell communication, and that includes molecular messaging. And that molecular messaging also has a tremendous effect on cell fate. And we're going to see that in a subsequent slide. Lipid would be a pathway. Of course, I love pathways, right? So you're going to have to see these. You have to put up with them. Besides, I think they just, picture tells 1,000 words, right? And I'm probably saying thousands of words, but the picture is going to say more than I can. Anyway, lipids also modulate directly gene expression, membrane dynamics, and metabolic flux. So lipids are all over the place in terms of cell fate. All right. So here's isoprenoid biosynthesis. I know this is just a review for most of you, but we start off with this hydroxymethylglutyryl-CoA, which actually comes from acetate. That is acetyl-CoA. And just to remind everybody that statins, the drugs that are used to block de novo cholesterologenesis, because that could be one of the end products, there it is down there, blocks right here at this HMG-CoA reductase step. Okay, so all these other intermediates in isoprenoid metabolism are also blocked when you use a statin. Now, I've always argued in all my advanced graduate lipid biochemistry courses that I created and taught, is I don't see how statins don't have some kind of pleiotropic long-term effect on human health, because we're blocking this entire pathway. And you can't make these intermediates from breaking down cholesterol in the diet. So I just don't see that there isn't going to be long-term effects of these prolonged chronic uses of statins. And it could be epigenetic factors that right now are happening that will be transferred meiotically to germline transmission recipients. But that's another story. Anyways, just to see the mevalonic acid is converted to dimethyl allyl pyrophosphate and isobenzyl pyrophosphate. And you start building up isoprenoids just by using these C5 compounds. Geranyl, farnesyl C15, geranyl C20. I also have talked about this in previous talks. Now watch this. This is really important. These two pathways, see this red star here I'm using, the one that makes carotenoids and then therefore retinoids, and the geranyl pyrophosphate utilization to make the phytyl pyrophosphates, such as chlorophyll, of course, that's the green pigment in plants, tocopherol, also found in plants, which is where we get vitamin E, and phytoquinone, also found in plants, where we get vitamin K. You see here that this pathway and this pathway are essential for us to take in the diet. We get vitamin A in the diet as usually carotenoids, right? Or some people actually take vitamin A in preformed vitamin form. Also, there's vitamin E as tocopherols, and there's vitamin K. So these are three essentials. We don't synthesize. These two pathways are not known to us. We do not obtain them. We have all the other ones, though. So, of course, we make cholesterol, and cholesterol makes bile salts, steroid hormones, and vitamin D. Boy, is that an important hormone. And also, this oligoprenyl PP makes plastoquinone and ubiquinone. Of course, that's really important, for example, in the electron transport chain. If that's not enough, we also make polyprenyl pyrophosphates, such as dolichol, and dolichol pyrophosphates are used to make glycoproteins. And many of these cell proteins that are made in the cell that are secreted, such, for example, let me think of one, like the immunoglobulins, right? Those are all proteins that require dolichol pyrophosphate to synthesize the glycoproteins in the endoplasmic reticulum for packaging into the Golgi apparatus and eventually to leave the cell via the plasma membrane. So that is a major pathway, right? Wow. All right, now I'm sorry about all of this, but you got to do it sometime, and now we're doing it. So phosphatidic acid, which is nothing more than taking glycerol and adding 2 fatty acids to the 1 and 2 positions of it, and then leaving the free phosphate, is metabolized further to all of the glycerol phospholipids we get in the cell. So that includes phosphatidylserine, phosphatidylethanolamin, and after adding 3-methyl groups, methionine and samethionine, phosphatidylcholine. So PC, PE, and PS are really important membrane phospholipids. You also make, by using the sugar inositol, phosphatidylinositol. Now, that's a major signaling compound, as well as an actual membrane phospholipid. Okay, so there's multiple pathways. You can kick up methanolamine and choline to make these pathways. Okay, so there's salvage pathways, or there's so-called nucleotide pathways because they use nucleotides, or directly just adding, or first of all, decarboxylating and adding methyl groups here. Because remember, serine is ethanolamine after decarboxylation, and ethanolamine is choline after trimethylation. Okay, this is all Kennedy pathway, named after Eugene Kennedy, who discovered all of this, partially when he was working at the University of Chicago in the 60s. All right, now, besides the phospholipids, we also have the sphingolipids. I didn't, I know, thank God, right? I didn't show you the sphingolipid pathway today because I'm going to really get into that later when we talk about ceramide and programmed cell death. But I just want to show you that when you have phosphatidic acid, like in the previous slide, remember that it's 2 fatty acids in the 1 and 2 position. You can take from that, you can remove from phosphatidic acid any of those fatty acids, and you can get back to having acyl or lysoacyl phospholipids. Okay, so and that includes diacylglycerol. Now, directly from glycolysis, you get dihydroxyacetone phosphate. You can take dihydroxyacetone phosphate, add a fatty acid from fatty acyl-CoA, and make acyl dihydroxyacetone. Now, this structure can go on to make another important class of glycerol-containing lipids called the plasmalogens. Again, right? Thank God I'm not showing you that. Really important phospholipids in the body. We're not going to talk about them today. I'm just giving you like brief, broad strokes what we're getting into here. Anyway, via the activity of glycerol 3-phosphodehydrogenase using NADH to reduce the ketone to the alcohol, you have glycerol 3-phosphate. Now, that's the backbone of the glycerol phospholipids. So you start adding fatty acids, you make lyso, that means it only has 1 fatty acid. You add another fatty acid, you get phosphatidic acid. Remember I told you that you can get there from here. Phosphatidic acid directly to make those phospholipids. We just saw that pathway. But dig this, look at this. Phosphatidic acid can also be used to make triacylglycerol. This is storage depot fat. So you have all the phospholipids, you can make plasmalogens, and the pathway is not shown, you can make sphingolipids. Likewise, you make the storage depot fat. So this is all going to be linked. This is back to bioenergetics, but it's also the storage of carbon for lots of other purposes. For example, making new phospholipids. So this is a really complicated nested set of pathways. I understand. And so today we're going to do it really easy. We're just going to look at this paper by Adrian et al. Published in one of my favorite journals, Lipids, published very recently this year. So let's take a look at what they're talking about. Okay, let's examine what this paper is. Let's get embedded in it. Cardiac hypertrophy, which I've again talked about in previous Verev Med video lectures, obtains via adaptation to elevated workload. That means you work out more, your heart gets larger, okay, in layman's terms. Now, that hypertrophy can be physiological, which means it's a good thing, okay, because you're adapting to elevated workload, or it can be pathophysiological. It's pathophysiological, this is usually a very bad news to people because it can lead to heart failure. So athlete-associated hypertrophy is usually considered pro-adaptive to performance while they are active, as, say, runners or pole vaulters or swimmers. But there's a pathological response that can occur both in athletes and non-athletes a lot. And in fact, that can lead to sudden death, sudden cardiac death. And that's associated with hypertrophy, actually. So pathology includes things like aortic stenosis, the associated hypertension with that, and that, as I said, can lead directly to heart failure. Again, go back and look at some of my previous logins for video lectures on that subject. Now, acyl lipid fatty acids are a major biofuel for the heart. Okay, the heart works on the oxidation of fatty acids more so than on glucose oxidation. You get more energy from the oxidation of fatty acids than you do glucose. And also, the heart has a direct way of isolating and obtaining fatty acids from the blood. So this is all a very important way that you keep the heart constantly beating throughout life until the day you die, right? Or unless you have heart failure or some kind of ischemic heart attack. So actually, fatty acid beta oxidation, okay, which generates NADH and FADH2, which can be reoxidized in electron transport chain, and then through chemiosmosis, proton motive force, generate ATP via complex 5, of course, in electron transport chain, making ATP, right? That accounts for about 80% of the output of the heart. Okay? So the major biofuel are fatty acids. Fatty acids or acyl lipids. Now, maladaptive myocardial hypertrophy is characterized by decreasing the amount of fatty acid being used and increasing the amount of glucose. Now, isn't that interesting? As you increase glucose oxidation and you diminish fatty acid oxidation, this is one of the biochemical signatures of maladaptive myocardial hypertrophy. So see, that's where metabolism comes right back in. So this paper wants to answer a couple of questions. First of all, it wants to ask, do short-chain fatty acids result in hypertrophy? Now, why short-chain? Because short-chain fatty acids, if you will recall from the last lecture I gave, is the major end product of de novo fatty acid synthesis, which is what we carry out, right? Humans carry this out. The most common short or actually medium-chain fatty acid that is generated in humans is palmitic, C16. I call that medium chain because it's C16. Short chain would be more like 12 and 14. Long chain would be 18 and above. So, however, they're going to be looking at short chain fatty acids here and medium chain, so C14, C16 specifically. And they're going to be looking at whether or not short or medium chain fatty acids result in an alteration of cardiomyocytes. That is, whether or not you see hypertrophy. And another thing they want to know is that, does this relate to apoptosis? And in fact, is apoptosis involved in fatty acid alteration in cardiomyocyte biology? Because I just told you that you're going to be shifting from using fatty acids to glucose. You might say, well, why are we even worried about fatty acids anymore? It's not all a glucose story. Aha! That's where you get the mistake in. The mistake is that it doesn't mean the fatty acids go away. It means they're used for something else. Okay. There you go. All right. So here's first data. Now, they have this weird name for this. I don't know why they were allowed to get away with this in lipids. If I was a reviewer, I wouldn't have liked it. But this is like a collection of fatty acids, okay? And they have palmitoleic, myristate, and palmitate. Now, palmitoleic is just palmitic acid with double bond at the delta 7 position, okay? So this is just palmitic acid with one double bond, a cis double bond. So I make palmitoleic acid, 16:1 for those of you keeping track. Myristate is just C14:O, shorthand for a saturated 14-carbon fatty acid. Not common, certainly not at the level they're showing here, 6 times more than this. This is a very uncommon fatty acid in humans. So I don't know where they got this little mixture from. And then they have palmitate, which is, this is the ratio of what they call in CoFA, this mixture of fatty acids they're using throughout the study. So palmitoleic 1, myristate 6, palmitate, C16:O, 16. 16, that's funny. And 1 and 6. They were playing around here, I think. All right, so that's what they're doing. So here, now they're asking, see what we're looking at here. Now I'm going to finally have to put on my glasses. Sorry. We're looking at serum concentrations. So they're just saying, you add this, do you get more of this fatty acid? Yeah, you get more myristic acid, you get more palmitic acid, you get more palmitoleic acid in the blood when you add this to the system. Good, okay? Serum concentration. So adding fatty acids results in an increase in serum concentration of them. Duh. Okay, but you need to know this is kind of like fulfilling the necessary result window, right? Now, another really important thing here. What about cardiomyocyte area. This is their way of describing hypertrophy. And after 7 days of feeding this, they really don't see any effect. Look at that. See? So it doesn't look like adding this fatty acids do anything, okay? Now, again, this is all from this paper, just reminding you. And the next 8 slides are actually from this paper. I didn't continue to cite that reference. Now, take a look at this. Again, this is cardiomyocyte area. We already showed you that the control versus these fatty acids didn't have any true effect because of the big standard error. Control plus TAC, there was an increase. Now, what's TAC? That's inducing a transaortic constriction. Okay, so when you do that, remember, hypertrophy is caused by a lot of cardiac muscle use, contraction, right? So if you induce a transaortic constriction, right, that's like using the heart a lot, okay, in this model system. So of course you're going to get more cardiomyocyte area. This is kind of like their positive control for saying, when we look at cardiomyocyte area, that's like hypertrophy. See, now they also did where they did this constriction and they added the fatty acid, and look what happens. They don't get the hypertrophy. So this is kind of strange, right? So definitely there's a difference here, right? You got a little bit of an increase in this cardiomyocyte area from the fatty acid, a little bit, right? But you got a tremendously larger amount when you do the constriction. When you add that with the transaortic constriction, look at that, you drop it back down. So that means that somehow hypertrophy is being regulated by these fatty acids. That's their argument. So they don't like the fact that all 3 of them together don't seem to have a profound effect, and neither do I. So, for example, so what they did is they said, okay, let's take these fatty acids out and try them individually. So again, we're looking at this stand-in for hypertrophy, right? Cardiomyocyte area, control versus the use of the fatty acids. They say no effect here, okay? Now you have palmitoleic acid, that's a little bit less actually of this hypertrophy. Now look at myristic acid. Wow. Okay. So the myristic acid is C14 colon, as I said. It did, by itself, seem to pretty high concentration, by the way. You'd never see this being bathed in cardiomyocytes, but it did increase hypertrophy in the system. And also, of course, TAC resulted in small increase in hypertrophy that was not enhanced by COFA. That's what we saw here. So again, the important point is myristate did increase hypertrophy. Okay. So now that's interesting. Palmitic acid didn't. Palmitic acid is going to be the far more common fatty acid anyways. For example, during obesity. So you're trying to link this back ultimately to diseases associated with dyslipidemia. Okay, but they never quite get there in the paper. I just wanted to remind you that's why we're doing this. Okay, again, now we're looking at apoptosis on the y-axis, increasing as you increase up the scale. And you're looking at all these different treatments. There's control. BSA is just a carrier for the fatty acids, okay? And the different concentrations of BSA, no effect on apoptosis. Look at this. No effect when you add this combination of 3 fatty acids on apoptosis. But when you increase the amount of it, you start to see some statistical increase in apoptosis. Okay, so that's kind of interesting, right? Because we also saw this when we split out amongst those fatty acids when we were asking for what? When we were asking for hypertrophy. Now we're looking at apoptosis. And I remember apoptosis is another fate of the cardiomyocyte. Okay, so hypertrophy can occur, but sometimes hypertrophy can be associated with apoptosis, a particular kind of apoptosis called necroptosis. That means basically necrosis, not good for the heart muscle, right? But anyways, here, look at this. Palmitic acid seems to have an effect more than the combination on apoptosis. Some increase at 50 micromolar, boom, wow, the biggest increase in apoptosis at 100 micromolar palmitic acid. Now, check this out, okay? ACAR, which is actually a nucleotide, okay? And ACAR is known to activate AMPKase. Now, again, I talked about AMPKase in previous lectures. I can't go over it again now. It takes too long. And people already complain about the fact that these lectures are too long, people I won't mention. So what I'm going to simply say is that ACAR activates AMPKase. And the AMPKase is that enzyme which basically shuts down anabolism and turns on catabolism. What kind of catabolism? You make more mitochondria, and you increase the amount of beta oxidation. Now look at that. When you add Acyl and you turn on AMPKase, you inhibit cell growth and mitochondrial apoptosis. Look at this. Even in the presence of the palmitic acid at the high concentration, Acyl blocks that increase in apoptosis. Okay, this is one of their like non-fatty acid controls. Of course, if you add reactive oxygen, you're going to get more apoptosis, right? Because it causes the cell to kill itself, because you have a lot of toxin in there. Now, this discussion here is really important. This is from the paper we're discussing in lipids, but this is what I found in the International Journal of Oncology, came out just about 11 months ago. ACAR works via AMPKase dependent peroxisome proliferative activator receptor gamma, okay? Coactivator 1A. Okay, that's PGC1A for those of you who don't want to say the whole name, okay? It also works through a mitochondrial transcription factor A. Now, I want you to keep that in mind, okay? So we're going to go back, we're going to look at this PGC1 alpha. It's going to be a transcription factor, and that's how AMPKase is going to transduce its signal. Okay, so in order for you to get this whole picture, you gotta go into the details. Now, I noticed that these days, the current repertoire of language out there, when they want to say something gets really complicated, is they say, "I don't want to get too much into the weeds." Well, I think that's really a bad way to talk about being complicated in your thoughts or in your language, because the weeds, remember, weed is specifically a plant that's growing in the wrong place. So getting in the weeds, you're into where there are plants growing in the wrong place. It doesn't work logically. I would say, rather than getting into the weeds, I'm saying getting into the molecules, okay? Because once you get into the molecules, that's when you start getting into the complexity. You see? It's a much better term. I recommend everybody now say that rather than saying getting into, I don't want to get into the weeds, say I don't want to get into the molecules. Because you see it works every time. And it will make all your friends be super impressed. All right, so I want you to keep this in mind about PGC1 alpha. It's going to come back to it. You can better remember. Okay, now let's go on through the data, continuing with the data. Look at this. Now, we're looking at, AMPKase is an enzyme that phosphorylates other proteins. One of the proteins it phosphorylates is our old friend, acetylcarboxylase. Old friend because I just talked about it in the previous lecture. Go back and look at that one. It's really important. It's the groundwork for this one. Acetylcarboxylase is that enzyme which takes bicarbonate and adds the carbon to what? To acetate, to make malonyl-CoA. Now, that is actually the first rate-limiting reaction for de novo fatty acid synthesis. Once you make malonyl-CoA, you are now detailing that carbon for lipogenesis. Mal-CoA is the substrate for lipogenesis. Mal-CoA is the substrate for lipogenesis, okay? Very important. So now look what happens here. This collection of fatty acids induces the phosphorylation of ACCase, but palmitic acid doesn't. Now remember, we already saw the palmitic acid has big effect on apoptosis, but no effect on hypertrophy, right? We saw that it had an effect on apoptosis. Okay, that's curious, right? Now we're seeing that when you take all these fatty acids, you also get an increase in the phosphorylation of acetylcarboxylase. What that mean? That means that you're shutting down the activity of the carboxylase. That means you're decreasing the concentration of cellular malonyl-CoA. Now, this paper doesn't do any justice to the fact that there are two forms of the carboxylase. One of them is associated with the mitochondrial membrane, and that's the gatekeeper for control of carbon flux for beta oxidation. And the other one is out there in the cytosol, which is committing the carbon to fatty acid synthesis. So I have a little bit of a problem with that, and I'm sorry for mentioning it because now I'm sure you're getting more confused, but just so you know, there are two forms of acetylcarboxylase. Only one of them actually contributes to the regulation of beta oxidation. They're acting like this pool of phosphorylated acetylcarboxylase is good enough to say that when you have phosphorylated acetylcarboxylase, like here, okay, right, and here when you use the ACAR, which activates the AMPKase, right, that you get an increase in this. Now we know that the AMPKase does that. So adding ACAR, even in the presence of palmitic acid, should definitely, because it's two kind of different pathways, presumably, should definitely cause a phosphorylation because that's a substrate for AMPKase, and ACAR activates AMPKase. So you get more of that. So basically what that means is when you phosphorylate, make less Mal-CoA. Less Mal-CoA means you can beta oxidize. So see how this fits into bioenergetics, switching back to fatty acid utilization. Fatty acid utilization. When you see that increase, you're increasing fatty acid utilization. Yeah. Palmitic acid actually prevents that by itself, which is curious because that's a common fatty acid for oxidation. So we'll get into this later. We'll get into the molecules. Okay, so ACAR phenocopies COFA. See, when I say phenocopies, I mean it acts just like it when you add it to the system. Okay. So the phosphorylation of the carboxylase prevents Mal-CoA from blocking beta oxidation. Therefore, QED, COFA promotes beta oxidation via AMPKase phosphorylation of the ACCs. Now, that's what they say. And then I go on with my nice, red-colored text, question mark. That's a paralogism. That's beyond what we can say with the premises. We don't know that that's how it works. We're just saying that's how it works. There'd be lots of other ways it's working. I would argue after going through this paper that they're not probably, at least they haven't convinced me. They haven't proven that. But that's what they're saying at this point. So I'm going to call that a parallel, beyond logic, beyond what is given with the premises, beyond what can be obtained by the premises. Careful not to conclude that way. Now, I'll go back to discussing logic later and talk, of course, so I know you're going to really enjoy that. Now, palmitic acid or ceticolinol fails to inactivate the carboxylase. See here? Doesn't inactivate it. And therefore, beta oxidation is inhibited, and that leads to apoptosis. That's another thing they suggest. Again, but what about the control? So here you don't have any palmitic acid, but you also don't have much phosphorylation of the carboxylase. So why is that? Okay, now we're running into some issues here, okay? So why is the resting state have less of this phosphorylated form? That would suggest that there's more of the non-phosphorylated form. And that would suggest that because there's less of that you would have more Mal-CoA, and therefore you'd be blocking beta oxidation. And I don't know if that serves their purpose. So see, you got to look carefully into the molecules to determine whether or not their broad sweeping generalizations always work, right? Or their propositions shouldn't be treated as conclusions. All right. So AMPKase, let's just take in total what they're trying to say here, and 16-colon-O, fatty acid, work in opposition to sulfate, where it looks like palmitate, and remember it enhanced apoptosis, and we showed you that the AMPKase actually shut that down, and maybe it has something to do with the switch in biofuel. We already know that, biofuel switching, when we know about hypertrophy. And remember, hypertrophy is different than apoptosis, but apoptosis is what can happen to cells in the hypertrophic heart, okay? Lots of cells that aren't able to maintain their active metabolic regimen are going to apoptose. So they're poised to after all this hypertrophy. That's the point that I also need to make. All right. Further with the data. By the way, these are all Western blots, okay? These are proteins. And you actually have antibodies to the phospho form and the non-phospho form. That's how they do this, right? Sure. Now, look at this. AMPKase alpha is phosphorylated by this collection of fatty acids. That's good. This is just the cell line they're using, okay? Cardiomyocyte cell line H9C2, human, see? Now, okay, cool. This is what you hope to see, that the fatty acids are working through AMPKase. That's why their argument, they think, holds. Now, that doesn't mean that it's not happening. It also doesn't mean that something is happening besides this. Remember the story of the ACAR, okay? And how it works. And we're going to see more detail about that in a few minutes. So, okay, we're getting phosphorylation. This is all like good. This is all like filling their hypothesis, right? Saying, well, you phosphorylate AMPKase. When you do phosphorylate that protein, you turn it on, unlike the carboxyls where you turn it off. Of course, you know that. Now, all right, so that, and you see this in the increase in the amount of phosphorylated form of the protein. Now, what else they showed, though, and this is a good control, and I really always like to see it, what about just the total amount of this protein? No effect. So it's not altering the amount of the protein. So it's not altering de novo synthesis of AMPKase. Okay, so no transcriptional translational control by the fatty acids. That's interesting. Of course, it's only a 7-day experiment. Still, that should be long enough to affect expression of genes. But also, it doesn't look like degradation is modified by it. That's also important to me. Okay? So no de novo synthesis of the protein enzyme, only an increase in its phosphorylation means an increase in its activation, which is all post-translational, which is good. So a combination of fatty acid enhances AMPKase phosphorylation, but not protein expression. Now, this is a complex slide. I'm sorry, but help, bear with me. I was going to say help me with this, and you'll help me by thinking through it. Okay, now I want you to get an idea about this AMPKase inhibitor we're going to be talking about. We're going to introduce a new compound. We had ACAR, which activates the AMPKase. Now, like all good scientific papers, if you can activate something, you can have something to knock it back. Remember, knock it up, knock it down, knock it up, knock it down, look at a phenotype. When you see it again and again and again, structure, function, structure, function, structure, function, you get an idea that maybe you are actually pushing the button on, off, or a little bit more, a little bit less amplitude modulation on a given target. Okay, that's why they're now got it. Now you're utilizing an inhibitor of AMPKase. Now, I didn't like what I didn't know about compound C. Okay, I'm not going to tell you all about it because it's quite detailed, big body of literature, which I did read. I'm just going to go to this one paper. This is Molecular Cancer Therapy, published now 3 years ago, almost 3.5 years ago or so, in 2014. Okay, now the AMPKase inhibitor, compound C, is a potent AMPKase independent anti-glioma agent. Now look at this. That's the name of the paper. They're looking at this inhibitor, but they're saying even though it inhibits AMPKase, the way that it blocks glioma, glioblastoma, that is major form of brain cancer, is actually independent of its effect on AMPKase. Now that's interesting, right? Because this whole paper here, this is from our paper. This is from these words are from this paper, okay, or my writing of it. Now check this out. Okay, it inhibits the AMPKase for sure, compound C, but this mode of action is not how it kills cancer cells. Why would shutting down AMPKase kill cancer cells? Okay, briefly, quickly, even though I talked about this again many times in the past. If you have a cancer cell and it's not getting fed enough fuel, one of the things the cancer cell needs to do in terms of stress is be able to go through an autophagic response, or be able to just turn on catabolism rather than anabolism, because anabolism costs a lot of energy. So some cancer cells aren't just proliferating. In fact, the core of the tumor may not be proliferating at all, right? The core of the tumor is just actively taking down cells, burning up carbon from the rest of the whole body, right? So blocking AMPKase in cancers can be a good thing because AMPKase, remember, is the catabolic response. It's there to make ATP. So if you don't have ATP, you cannot get some of the other cells in the tumor to proliferate. You see, right? So AMPKase annihilation, AMPKase blockade is something that people like to think about could be useful as one of the many pharmacotherapeutic arms in attacking cancers, especially the particularly grossly aggressive forms of glioblastoma. Now, okay, so it inhibits the AMPKase to show it in this paper, but it's not how it kills the cancers. Rather, they found that what the inhibitor compound did was it activated an entirely different pathway, the calpain-calthepsin pathway, which actually works to inhibit AKT and mTORC signaling. And that provides a cell cycle block at the G2M phase, and then that induces necroptosis and autophagy. Now, necroptosis is nothing more than apoptosis, but it's necrotic, meaning that the cells lyse and they fall apart and they leave a lot of organic compounds behind so they can cause an immune response, okay, which isn't necessarily very good, particularly in the brain. And so don't know how useful this is going to be in the long run, okay? But look at what it's doing. It's not using AMPKase's activation of beta oxidation. It's using this inhibition somehow, inhibition of the anabolic pathway. Right? So that's like taking it and turning it on its head. Okay, so that's very curious. Probably via feedback inhibition. So I'm sure it's still working through the AMPKase modal operator, right? But I think it's via some kind of background trapdoor feedback inhibition that it's now shutting down anabolism. That's what's probably happening here. And remember that necroptosis, again, it's a new term, but you can get where it comes from, is an inflammatory mechanism of programmed cell death, which I just said. Now, okay, so that's all from that paper. So I just want you to know that what they're using, so this is now we're back to our 2017 paper, in lipids, remember. Go to the next 8 slides are going to be that. This is data from it. Here they're introducing compound C. They're saying, look, oh, wow, we got this AMPKase inhibitor, and look what happens. When we are looking for phosphorylation of AMPKase, we get a big increase when we add myristate. Myristate, remember, was also the fatty acid that was involved in, what was it? Oh, yeah, hypertrophy, right? Here's your control, right? Here is your myristic acid, that's C14:0. You get an increase in phosphorylation. It looks cool. Look, and when you block the enzyme, you block, right? So this compound C is supposed to block the activity of AMPKase, it inhibits AMPKase, and so therefore you're blocking the phosphorylation of it. Okay, so that's showing you're debilitating the production of the active form of the AMPKase. So what they say is the authors, they say that myristate, that's 14:0, enhances AMPKase activity. Now, is that justified? Okay. Now, given this literature here, I would say no. It could be, but I wouldn't say that there is any real reason to say. Again, it's a paralogism. It's going beyond what the data will allow. Okay. Definitely doesn't mean that this is a direct correlation. It just means that somehow this compound is knocking back the phosphorylation of this enzyme. But it could be because it's inhibiting all these pathways, and that could also diminish the amount of phosphorylated form of AMPKase. Now, why would that be? Maybe because when you block this all down, you have to decrease the amount of phosphorylated activity of this because the steady state level of AMPKase is already sufficient to carry out what needs to be done, which is programmed cell death, and not what AMPKase would like to do, being anthropomorphic there, which would be more like maintenance, right, and repair and senescence at worst. All right, more on the data now. Again, the same paper. Again, these are all Westerns. I just reminded myself. Western blots are just looking at protein, right? They're immunoblots. I'm sure many of you know this. And if you don't, it just means that what we're looking at here are bands of proteins that are detected by using an antibody to detect the protein in the cell. Okay, that's a Western blot. So what we're looking at here now is another protein, another possible protein involved in this entire pathway signaling process that they know is important. And that's the fat CD36. Now, again, I have a really excellent talk on that pathway, okay? What fat does and CD36 does in skeletal muscle. Now we're in cardiac muscle. Just want you to go back and look at this sometime. Basically, CD36 is involved in taking fatty acids from lipoproteins and getting them into a cell so that they can be metabolized, either synthesized triacylglycerol, like in adipose tissue, or for beta oxidation, or for other utilization of fatty acid in the cell. So that's what CD30 is. It's a receptor protein on the plasma membrane of these cardiomyocytes, which allows the uptake of fatty acids. Now look at this. Now that protein, okay, is increased by adding palmitic acid, right? It's not, and it's also increased some by the total package of collection of fatty acids. But when you add ACAR, which activates the AMPKase, you reduce this amount of this translocating fat CD36 plasma membrane receptor for fatty acids in the cardiomyocytes, back down to control levels, at least statistically. So that is important. Again, it shows that AMPKase activation somehow blocks this. Now, blocking this means what? It means it's shutting down the movement of fatty acids into the cell. Okay, so again, AMPKase isn't necessarily involved in bringing fatty acids in. It's involved in oxidizing what's there, utilizing what's there, right? All right. It's a curious step here. Now, here's another protein called ADRP. That's the protein, basically, it's called adipose differentiation-related protein. You find that was first described in the adipose. That's the protein that basically gets involved in setting you up for making triacylglycerol, the storage lipid, or what they're called lipid droplets in the cell. Very important. Skeletal muscle, right? It's a storage depot of triacylglycerol, which can be utilized after hydrolysis of those fatty acids from that glycerol backbone for energy. So what this shows is that palmitic acid increases fatty acid uptake, through the CD36 receptor by increasing its expression, and enhances lipid accumulation via the adipose differentiation-related protein. So you accumulate those fatty acids, you accumulate them, you don't use them. That's what this shows you. But increases the expression of ADP-RP, increases the expression of CD. That's what they're arguing. And that when you activate the AMPKase, you reduce that. You reduce the amount of triacylglycerol production and reduce the amount of fatty acids coming in. Okay, not quite down to control levels, but back down to levels that are much lower than what palmitic acid afforded. So remember that Acyl activation of AMPKase blocks the effect. That's what we see. Now, finally, almost, palmitic acid enhances the SAPK, okay, it's a stress-associated protein kinase JUNK pathway. And now that pathway, the SAPK JUNK pathway, JUNK is a form of a SAPK, remember, stress-associated protein kinases, that is all linked to programmed cell death. So palmitic acid increases programmed cell death and appears to be blocked by activation of AMP via Acyl. Okay. Now we already noticed this when we're looking at what? Total apoptosis, the palmitic acid increased apoptosis. This is getting down to the molecules, right? Moving amongst the molecules. These are the molecules involved in triggering apoptosis, this kinase pathway. And indeed, palmitic acid, even at the low concentration, 50 micromolar, I wonder why they didn't have the 100, by the way. Maybe because they didn't see an increase in activity when they added more of that fatty acid, which is possibly problematic, unless maybe you're maxing it out and you just go plateau. Anyway, you can see that you increase the amount of these enzymes which are involved in apoptosis. And then when you use Acyl, AMPKase, you block that, so you're blocking apoptosis. So this is their final paper. This is their final slide, excuse me. And I took this directly from their paper. It's a summary slide. And there's basically showing you that phospho-AMPKase is the main regulator of cellular energy homeostasis. Now let's look. They're claiming that hypertrophic growth, okay, somehow leads to the production of myristic acid. Okay, now they get this from previous work they did, but there's no real good indication that you get a lot of C14 colon O because of hypertrophy. Okay, because that's not the common de novo synthesis product of fatty acid synthesis. But anyways, they're claiming that hypertrophic growth causes this increase in myristic acid. That causes an increase in the phosphorylation of AMPKase, which causes the phosphorylation of acyl-CoA carboxylase. When you phosphorylate acyl-CoA carboxylase, you shut down the mal-CoA. When you shut down the mal-CoA, you increase beta oxidation, okay? But because of that increase in beta oxidation, this slide is messy, right? You block apoptosis, okay? So that's what they're trying to show here. And you block lipid accumulation because you're using the fatty acids. And they're claiming that the collection of fatty acids does the same thing, so they're arguing it must work through this pathway because they don't put a question mark here. Yes, see. The same myristic acid is the fatty acid agent that's responsible for the way the collection of fatty acids work. Okay, that's why they're showing this. Total global effect, blocking apoptosis lipid accumulation. Now, they're claiming hypertrophic growth is not involved in palmitic acid increase. They say they don't think that happens. They think that the palmitic acid is coming in from increasing the amount of this receptor-mediated endocytosis of fatty acid and lipoproteins. They think that that's all helped by the ADRP pathway, right? So if palmitic acid increases the expression of CD36, increases the expression of ADRP, that increases lipid accumulation as lipid droplets. They also showed you just a moment ago that they increase the amount of stress-associated protein kinases. That's going to do two things. They say it's going to increase programmed cell death and increase lipid accumulation. It's going to be a bad thing after, subsequent to hypertrophic growth. Okay, so they're trying to link the two things together. First, you get hypertrophy, cardiac hypertrophy. Then you get apoptosis, and that leads to heart failure. That's the point they're making. Okay. So the way they put it is the CoFA activates the AMPKase, lowers cell apoptosis. Myristic acid activates the AMPKase, and it induces hypertrophic cell growth because they had shown that previously, and they show it today. All right. So they say the palmitic acid increases the expression of the PAT-CD and ADRP, and that's all independent, you see, of AMPKase. Now that's curious they're saying that, because remember several slides later they were talking about how palmitic acid seemed to have some kind of nuanced effect on AMPKase. So they're trying to like remove themselves from that one of the subsets of the null hypothesis. So this was not, it didn't set with me very well. It wasn't a savory response to me as a lipid biochemist, even though this is in the journal Lipids. So I wanted to like tear this apart a little bit. And the way that I do it is I go to the literature. So so I asked, conclusion not found? Conclusione et non invenne? That means it's the conclusion not found. The conclusion that this last slide here, right? The AMPKase pathway is blocking this. Palmitic acid is activating this, and that's what results in having the apoptosis after hypertrophic growth, and that causes then the problem with heart muscle necrosis, right? Necroptosis, and also ultimately heart failure. I'm saying, do we really find that? And here's what I'm asking. Where do the palmitic acid, palmitoleic acid, and myristic acid synthesize, and how is it introduced to cardiac muscle? We just don't make a lot of that, okay? There's some, but certainly not the ratios they were talking about in that COFA. And so and then pure myristic acid, no way is there that much of it in the cell. So it's curious that they're trying to push this, okay? And they're pushing it because they found that's how it worked, okay? Now, I would say, okay, given the data, and I'm going to back it up with some looking at some literature, that it's more likely that the palmitic acid is esterified to CoA cytosolically and therefore removed from beta oxidation in preference to make the tag. And because of that, that can be pro-apoptotic, and maybe the pro-apoptosis isn't so much with sequestering the acyl-CoA, but maybe because the palmitic acid is used for sphingolipid metabolism. Now, I didn't show you how you make sphingolipid. I told you I was going to spare you of that. But sphingolipids have, to make the sphingosine base, you need palmitate. So maybe increasing palmitate in these cells increases sphingosine metabolism. Sphingosine metabolism ultimately leads to ceramide. Ceramide is a sphingolipid, which is directly linked to programmed cell death. Now, they didn't look at that. I would have. Okay. Now, anyways, if that's at all possible, this is just a question. It's just hypothetical deduction on my part. Now, this could generate, though, having this fatty acid going to the tag, okay, which is what they're kind of trying to argue, but they didn't flesh out how it worked and how I'm doing this again. But both of these things, putting the palmitic acid in triacylglycerol, increasing the amount of stored lipid in cardiomyocyte, and perhaps being pro-apoptotic by increasing sphingolipid ceramide synthesis, those things could generate a competition between the mitochondria and the cytosol for fatty acid. So you're not getting fatty acid into the mitochondria. That paradoxically could actually activate the carboxylase because the cells start to starve, because remember, they want to be beta oxidized and they don't want to use glucose. You don't want to use GLUT4 here to bring in more glucose because you want to use more fatty acid. So paradoxically could activate the carboxylase, activate fatty acid synthase. And these, by the way, are the direct substrates for the AMPKase, right? So AMPKase is going to shut down the carboxylase by phosphorylating it. We saw that in their data. It's going to shut down the expression of the enzymes in fatty acid synthesis because that's anabolic, that's lipogenesis. So I see more of that if the palmitate gets shuttled into triacylglycerol oil droplets in the cardiomyocyte, and possibly also as a subroutine into sphingolipid synthesis, which is directly going to be involved in programmed cell death and the necrosis type, right? The necroptosis. That's looking at the literature and knowing what I know about lipid metabolism. That seems to be more of the more possibly what the actual mechanism is. So in order to go to examine that at all, I did a little bit of a dialectic. Let's take a look at some premises we can find in the literature, and let's finally come together with an ultimate conclusion for today. Now, this paper comes from Anti-Cancer Research 2016, and the title kind of says it all. Inhibition of fatty acid synthesis induces apoptosis in human pancreatic cancer cells. And what they show is if you block the acetyl-CoA carboxylase, you get programmed cell death. If you add back palmitate, you rescue these cancer cells from this fate. Now, that's weird, right? Because they're saying blocking acetyl-CoA carboxylase, which seems to be what the AMPKase does, in this system, you're getting programmed cell death. Now, that doesn't surprise me, because if you don't have fuel, right? If you don't have fuel and you inhibit fatty acid synthesis, you're going to cause these pancreatic cancer cells to die because you're not taking in enough glucose. And when you add just bioenergetic substrate palmitic acid, you rescue them from apoptosis. So here, it's totally working a different way, right? Here, the fatty acid is actually fuel for the cancer. It's not causing apoptosis because the palmitic acid is not being used in this system to make triacylglycerol, or probably also to make ceramide, that is a sphingolipid. So cardiac muscle and the ductal carcinoma of pancreatic cancer are different. That's what I meant at the very beginning about how you need to look at the different tissue types, and you need to look at the different biomedical situations, the whole picture of the biomedical situation. We're looking at a cancer, what kind of cancer, glioblastoma, ductal carcinoma of the pancreas versus hypertrophy in the heart, and asking what palmitic acid does. Even though the same players may be involved, the way that they function is differently. Okay? And what's wrong with that? Nothing. Okay? Because that's how the cell works. The cell is highly differentiated in its metabolic pathways and its feedback regulation. Now, take a look at this. Influx of palmitate enhances tumorigenesis, and that does so directly because it's a bioenergetic fuel. Pancreatic ductal carcinomas don't produce TAG, okay? So you're not going to shuttle that incoming palmitate into TAG, which in the last slide we said could be argued to cause apoptosis. So therefore, you lack the potential to increase programmed cell death by introducing palmitate in this system. Plus, they generate desmoplasmic film, okay? So that's what happens. So this desmoplasmic film actually induces hypoxia in the ductal carcinoma. And that switches metabolism anyway to glucose and glutamine oxidation, okay, rather than beta oxidation. So you're not going to be using these fatty acids for beta oxidation anyway. So, but if you add it back, okay, if you add enough of it back, you can overcome it. But if you induce hypoxia, which is what they did in this paper here in National Journal of Cancer, which was published in 2015, that's where I got this information, okay? So if you add palmitic acid to this system, okay, it gets blocked in its increase in tumorigenesis if you give it a shot of hypoxia, if you remove oxygen. That's because you're blocking beta oxidation, you see? Anyways, because the cell wants to move towards glucose and glutamine oxidation because the fatty acids can't get through because they have this desmoplasmic film. So there's actual structural differentiation which prevents the utilization of the palmitic acid. See, so you really have to get down into the molecules to make any kind of statements, and none of them are particularly bold. They're nuanced, but they're very sophisticated and they're very precise, and they're probably much closer to authenticity. So this is all distinct from hypertrophic cardiomyopathy, which I've been just saying. In that system, we sequester palmitate into TAG, and that induces apoptosis either directly, remember, I told you before, with the feedback regulation of the carboxylase and fatty acid synthesis, or perhaps via the sphingolipid ceramide pathway, which again, I didn't show you any slides of that, but believe this, that ceramide is involved in necrotic apoptosis, right? All right. So this kind of puts it together. Again, this is my formulation of the system, right? So acute hypoxic preconditioning prevents palmitic acid-induced cardiomyocyte apoptosis via switching the metabolic GLUT4 glucose pathway back to CD36 fatty acid dependent. Now, this came out, okay? This came out. It's still in press form. This is in the Journal of Cellular Biochemistry. This was accepted just, a month ago, not even, November of this year. Okay, this is December 7th. I'm speaking. This paper was accepted November 9th. So this is in the Journal of Cellular Biochemistry. Now look at the title again. Acute hypoxic preconditioning prevents palmitic acid-induced cardiomyocyte apoptosis. Because here, palmitic acid is again inducing apoptosis. Okay, because it's back in the cardiomyocyte. You see, it's not looking now at the pancreatic ductal carcinoma. And it does so by switching the metabolic GLUT4 glucose pathway back to, okay, so it prevents it because you bring in, the palmitic acid starts going back to bringing in fatty acids. That's what it does as well. Remember, that's what we showed it did. Yeah, it induces this whole pathway, right? Induces CD36, and remember the ADRP. Okay, so here's how you can put it together. This is complicated, but it's beautiful. And I'm putting it all together from all the data and also from the literature. Now, we know that this protein sirtuin-1, now that is a protein deacetylase. That's all you need to know right now. It removes acetate from proteins. When AMPKase is activated, okay, when it's phosphorylated, it is going to convert the SIRT1 to phosphosirt1, okay, with utilizing the gamma phosphoryl from ATP to make phosphosirt1. That's the active form of the enzyme. The active form of the enzyme is going to deacetylate this transcription factor, which I told you we were going to talk about. Finally, here we are. And that is PGC1 alpha. Remember, this is the peroxisomal proliferator activator protein, conjugate adapter protein involved in the regulation of what PPARs do, which is basically to turn on beta oxidation. So watch what happens. SIRT1 deacetylates that enzyme. The enzyme, when it's deacetylated, is activated and now can be translocated to the nucleus. When it's translocated to the nucleus, it increases the expression of PPAR alpha, peroxyproliferated activator receptor alpha, increases its expression. Therefore, it allows it to make a dimer with the retinoic acid receptor. That dimer acts as a ligand, acts as a transcription factor on making CD36. Okay, so AMPKase phosphorylates SIRT1, which deacetylates PGC1 alpha, which is translocated to the nucleus, which combines with RXR, that's the retinoic acid receptor, so you got the isoprenoi lipid there, right? Combined, this acts as a dimer transcription factor to now turn on the expression of CD36, allowing more fatty acids to come in. So this whole system, you see, is how palmitic acid normally induces cardiomyocyte acidosis by switching. You see, so that's the whole mechanism here. And this hypoxia, you see, right? This hypoxia, which is generated when you have an ischemic heart attack, for example, is going to stop all of this because you're not going to have oxygen for beta oxidation. So CD36 expression increases the influx of serum fatty acids as biofuel. It requires beta oxidation increase via the expression of another protein shown in this paper, which is the carnitine palmitoyltransferase 1 beta. And so that enzyme, or that's an enzyme, that protein involved in translocating fatty acid in the mitochondria, which is necessary for beta oxidation, and the CD36, both of those are transcriptionally activated by the PPAR alpha RXR transcription factor dimer. So, let's put this together. Fatty acids can both promote and prevent apoptosis depending on cellular environment and time. And we saw that. We're looking at versus ductal carcinoma, glioblastoma on one hand, or you're looking at hypertrophic cardiomyocytes on the other. Okay. Same metabolic regulation, that is, you're still using the same players, AMPKase, the PPAR, the GLUT4, because it's going to be involved in glucose uptake, and CD36 fatty acid uptake. All those same players, right, can result in what I call the pseudo paradox of contrary outcomes. Okay, they're contrary outcomes because it seems like they shouldn't work, but in fact they can work simultaneously. Okay, so remember the contrary. What does it mean? Okay, when we're talking about logic, contrary is not what? It's not contradictory. So contrary means both could be false, but when you have a subcontrary statement, both can be true. Okay. Now, a contradictory statement means the existence of one annihilates the existence of the other. So I'm telling you this is all a big pseudo paradox because you're just looking at contrary outcomes, which means that two things can happen. Okay, you can have two things happen at the same time, or neither one of them happen. Something else can be happening, and that something else can be happening has to do with that whole triacylglycerol, even sphingolipid metabolism. So again, using logic, categorical logic, and superimposing that on the data, you're able to um, to draw forth a set of conditions to explain what the data says holistically from a lot of other publications, and still come up with a framework of understanding of all these intermediaries in fatty acid metabolism and all the signal transduction through AMPKase and PPAR and the lipid accumulation, right, and glucose versus fatty acid oxidation. All of that comes together into subsumption, into making sense, right? But you have to look at the individual cases. And the way that you go about doing that, the way that your brain goes about doing that, is analyzing it by using logic and knowing that there aren't contradictions. So these are all pseudo paradoxes. So to unravel these biochemical, biomedical, what I'm calling now memes, so I'm saying there's a new meme out there, biomedical, biochemical memes, that requires careful experimental approach, like design your experiments correctly from the get-go. And to do that correctly, you have to now transform your logical interpretation of what experiments you're going to do and what your data says, and vetting that data against the literature by using this other very important system in philosophy known as dynamic logic. So dynamic epistemic logic is basically systematically dealing with real-time reevaluation of core beliefs. That's known as doxastics. Doxa means belief in Greek, and the general epistemic revision. So this is what your brain needs to be doing when you're looking at scientific literature. And you're not only looking at the literature that's published, you're also examining what to do next. So you're interpreting the data, you're evaluating the evidence, you're trying to synthesize it into a discussion, which is what I just did there at the end of this talk. At the same time, you have to be able to reevaluate your core beliefs, because all of knowledge has to do with beliefs, as well as has to do with truths, as well as has to do with justification, right? So if you believe a certain system to work a certain way, you have to be able to reevaluate that belief and pull back one level, move upstairs one level. And that involves dynamic epistemic logic. It's really important for scientists to do. So I'm going to end here today. Again, this is a picture I took in the Colorado Rockies. Beautiful, right? This is actually the Continental Divide between Wyoming and Colorado, where we were camping recently. There's our email address for the company, info@verifmed.com. Our website is there. Please come visit it. Please come visit it and contact us via our email address. Remember that what we are is we are scientists verifying published evidence in medical biosciences. Hopefully, I've demonstrated that today when I carried you through this discussion of fatty acids. This is my Facebook page. On my Facebook page, you're going to find posted all of my VeraMed video lectures, YouTube lectures. Okay. If you have a problem with finding those, contact me directly. Here's my email address directly to me, and I will make sure you get copies of all of those previous. Now they're up to maybe 30 different video lectures. Right? Quite a library I'm generating here in terms of hours online of trying to put together some of this data. Remember, what I'm doing in these talks, in these video lectures, is to demonstrate what we can do for you as a client. You come to us with a given physiological or biomedical condition or paradigm. If you're a physician and you want to know about drug-drug interactions, or you want to know what the literature is saying about how maybe surgery and drug combination or radiotherapy are involved in a person that has a heart condition as well as also is now presenting with a cancer. This is the kind of way I can synthesize the literature for you to help to come to some conclusions of what the literature is telling us, and to move forward with that information to then ask whether or not conditions can be afforded to move forward with the biomedical research program. That's me, and that's my email address. Please contact me. I'm always available, and even though my email sometimes gets overloaded, I will answer every single email I get. So I want to thank you again for listening to this, I know, rather long talk. It's a complicated subject. I guarantee you it's more complicated than what we did today. I bracketed off the whole lipid story for a reason, for example. But I want you to get an idea of how you can take given literature, the published literature, you can tune it to explain how that literature does or does not fit into the compendium of literature. So can you justify what the conclusions are of this paper from the justification conclusions of other papers by looking at the evidence and verifying it? And that's what I'm about doing. So all of these seminars are just basically calling cards to demonstrate what we can do. When we work with you, we work on an individual basis. We can get down to discussing things at various levels of topic, right? Various levels of penetration of the scientific literature based on acumen of understanding, all the way to being very sophisticated and having a very good background. And for example, bioenergetics and molecular genetics, or to a person that has absolutely no background in this, and I can still bring them forward so that we can interpret this data just by dealing with it with different sets of conditions, different nomenclature, and different, perhaps even syllogisms. So hopefully this was beneficial to you. Please give me feedback. It's always something I treasure. We will meet again soon. I've got 2 more lectures before Christmas. Again, this is Dan Guerra saying thank you very much for listening to me today. And as I've been saying, I want to say one last time, bye for now.